Hello and welcome to this video in the series on integrated management of cereal diseases. In this video we're going to start into the methods for controlling disease epidemics. In later videos we're going to get more into the practicalities of particular control methods. In this video we're thinking more about the principles of how the epidemic happens and what we can do to either delay it or slow it down. So I should warn you that this video may contain equations. If school puts you off mathematics as well as cabbage, then you'll find that you can quietly ignore the equations and all the rest of the logic should still make perfect sense. Okay, so let's get started. Hello. This is a video in the series on the science and practice of integrated pest management, where we're focusing on disease control in cereals. And this time we're focusing on the question, how can we delay and slow disease epidemics? And we're going to use the examples of Septoria tritici blotch and yellow rust in wheat as contrasting examples. But let's start by thinking generically about disease epidemics. This is a typical sort of hypothetical epidemic curve. If the disease is allowed to run its course, then through time it starts off, accelerates rapidly in an exponential phase, and then eventually follows this sort of S shape for reasons that we'll come back to in a moment, with severity increasing over time. If we're wanting to protect an individual leaf layer in the crop canopy, such as the flag leaf, then we need to delay or slow this epidemic to keep it at a very low level for the natural life of that leaf layer, which will be about 2 months. If we're thinking about protecting the whole of the upper canopy, then we need to slow or delay the epidemic for a longer period of time. So what are our options for doing that? So first of all, let's just think a bit more about the different phases of the epidemic before we think about what we might do to deal with those different phases. So if we just focus down in the little blue circle at the bottom left of the chart for a moment, the disease doesn't come from nowhere. It's coming from some spores arriving from somewhere else, which we're terming here initial inoculum. So in effect, the epidemic is starting from some small amount of disease coming from elsewhere, and we may be able to do something about that. The next phase, once that disease has arrived, is a phase of exponential growth in the orange outlined circle, during which the disease is growing very rapidly, potentially. But then what we can see is that if the epidemic is allowed to run, it actually starts to slow down and reaches eventually what we call disease carrying capacity, which is the upper limit of how much disease the crop canopy can support. Now, if nothing else constrains that carrying capacity, then what ultimately limits the epidemic is that the pathogen runs out of new leaf to infect. So more and more of the spores it produces are landing on previously infected leaf area, and that slows the epidemic down. But that's really no help to us because we can't allow the epidemic to get anywhere near the kind of level at which that starts to slow things down. But there may be other things we can do to affect the carrying capacity, and we'll come to those in a moment. So essentially what we've got are 3 options that arise from these 3 phases of the epidemic about what we can actually do to delay or slow the epidemic. So the first is we can try and shift the epidemic in time by delaying it. Second thing we can do is to try and reduce the epidemic growth rate, particularly in the exponential phase. The third thing we can do is try and reduce the carrying capacity of the epidemic and stop it reaching a high level, which also means it slows it down before it reaches that high level. Now, we can see in these sort of hypothetical examples, the control in each case is not fantastic, but if we manage to combine two or more of those methods of delay, reduced growth rate, reduced carrying capacity, then we can have quite a profound effect on the epidemic. So we're really looking to integrate different control methods to achieve these different effects on the epidemic in order to achieve that combined effect. So let's look in a bit more detail now about how each of these works. So we saw in the previous video about how to predict epidemics, what the main factors are that control epidemic growth. And those two things were the basic reproductive number, R0, which is a measure of the number of daughter lesions produced by each latent lesion in the phase when they're not competing for leaf area. And the second component is the generation time, which is how long it takes for the infection cycle from one lesion forming to a daughter lesion forming. And the basic reproductive number is determined by the probability that a latent period survives to become infectious, the number of spores produced by a new infectious lesion, the probability that one of those spores produced by an infectious lesion lands on a leaf rather than just dropping to the ground, for example, and the probability, if it does land on a leaf, that it manages to infect and produce a new latent lesion. So how does R0 and generation time work in their effects on the epidemic? So the first point is that the basic reproductive number R0, as we heard during the COVID pandemic, is very sensitive as to whether it's greater than or less than 1. If each lesion produces more than one new lesion, then the epidemic is going to grow exponentially. If, on the other hand, each lesion produces on average fewer than one new lesion, then the epidemic is going to die out. And we can see that in the chart on the right-hand side. And there can obviously be weather conditions in which R0 is less than 1 and the epidemic just does not develop. But in many cases it will, so we need to think about how to manage that. So let's think in a bit more detail. Let's imagine we start with a certain number of initial lesions. So this is our kind of initial inoculum. We'll call that S0 here. After one generation, we start with S lesions, R0. Suppose each lesion produces 2. Then if we start with one lesion, after one generation time, we've got 2. After a second generation time, we've got 4. After a third generation, we've got 8, and so on. And plant disease epidemics can occur at least that fast and substantially faster, as we're all aware. Now, two things come from this. First of all, we can slow down the rate of the epidemic by reducing R0. And for example, variety resistance and fungicides all affect R0. The other thing we can do is try and reduce the initial inoculum. So reducing S0. So if we imagine for a moment that our basic reproductive number is 2, that each lesion produces 2 new lesions, if we reduce the amount of initial inoculum by half, then we effectively delay the epidemic by 1 generation time, because having gone to a doubling of the number of lesions in 1 generation, it's back where it would have been in terms of the epidemic growth. If we can reduce the initial inoculum 4-fold, we'll delay the epidemic by 2 generation times. And if we can reduce the initial inoculum by 8-fold, we'll reduce it by 3 generation times. So that's the kind of theory of it. Unfortunately, that theory also actually works in practice. So this is data from a series of experiments that we did some years ago, where we manipulated the amount of yellow rust initial inoculum during the winter and into the very early spring, and then we left plots of a susceptible variety untreated to allow the epidemic to run. And we can see here exactly the delay in the epidemic. So the curves on the left-hand side, we didn't reduce the initial inoculum. That was just the natural background amount. And as we decreased from that level of initial inoculum by experimental treatments, increasingly we delayed the epidemic by 10 or 20 days. And that 10 or 20 days is buying time for us to manage the epidemic. And the other factor which can substantially affect the amount of initial inoculum, as we know for yellow rust, is the severity of the winter. That if we're lucky enough to get a really hard winter, it can knock back the initial inoculum by killing out the lesions in the foci in the crop, and that can slow the start of the epidemic in the spring, which also buys us time. And we'll come back to management options to achieve this later on. So that's the first option, to delay the epidemic. Second option is to reduce the rate of epidemic growth during this exponential phase. And as we've seen, that depends on the basic reproductive number, R0, and the pathogen generation time, and whether host resistance, fungicides, for example, and other disease control measures can all help to reduce R0 or extend the pathogen latent period, which increases generation time and slows down the epidemic. And then the third option we've got is to try and reduce the carrying capacity. And again, this doesn't just work in theory, this has application in practice. These are data from a series of experiments where we limited the nitrogen fertilization of a crop of winter wheat and then allowed an untreated yellow rust epidemic to develop on that crop. And we can see here the top curves are where the nitrogen inputs were high, and each of the curves below is where we reduce the amount of nitrogen applied. And we can see in the bottom case that the epidemic is actually running out of steam and stopping, despite the fact that at the point it was stopping, there was still lots of apparently healthy green tissue for it to affect. But it had run out of carrying capacity because the yellow rust has a high demand for nitrogen to be able to continue to grow. And that's also the case for other biotrophic pathogens such as the powdery mildews. So let's just think about our two contrasting pathogens and what the options might be for achieving these different methods to slow the epidemic. So Septoria tritici is a necrotrophic pathogen. It has to kill leaf area before it produces spores, and it can produce spores on dead leaf tissue. And that means that there's quite an abundance of crop residue left at the end of the season for it to produce ascospores on. And those ascospores are aerially dispersed by wind, and they can disperse and infect the new season's crop when it emerges in the autumn. So we'll see immediately that trying to control initial inoculum is going to be difficult. And the other thing is that although the nitrogen content of the leaf has some effect on the growth of Septoria tritici, the effect on Septoria is relatively small. If we contrast that to the yellow rust case, so yellow rust is biotrophic. It has to live on live leaf tissue. And that creates a bottleneck for it in between crops. So when we harvest one crop and sow the next crop, it needs a green bridge of live leaf tissue in order to be able to survive between seasons. So that's a limitation on it that may affect its level of initial inoculum going into the next crop. And the other problem it has is that it's really quite inefficient at long distance dispersal. So the urea spores that are produced tend to clump together because they're quite sticky into little lumps of spores, and it takes a lot of wind to dislodge them from the leaf surface and move them, compared to an ascospore of Septoria, which is actually blown out into the wind. And also, because these spores tend to be in little clumps, they can sometimes sort of fall out of the air more rapidly. So it's fairly inefficient at long distance dispersal, which is why we tend to see small numbers of foci in crops where a clump of spores has managed to infect, rather than a kind of widespread infection which we see for Septoria. However, in its favour, the short distance dispersal is really efficient. So plants rubbing together or spores moving a short distance in the air, less than about a metre or two, is really efficient, which is why those foci of yellow rust we see in the spring can extend and expand so incredibly rapidly. And the other point is that the nitrogen content of the leaf can have quite a strong impact on the carrying capacity of that leaf, as we saw earlier. So we'll have gathered from this there are quite different control approaches that are needed for the two pathogens. So if we think about methods to reduce the initial inoculum and delay the epidemic, crop rotation has no effect on the severity of Septoria. The preceding crop doesn't affect it. We see this in the DEFRA Pest and Disease Survey of commercial crops. There's no difference in the Septoria severity between, for example, wheat crops following wheat or wheat crops following a break. Whereas for yellow rust, because the volunteers from a preceding crop can be a source of green bridge, then crop rotation and preceding crop can matter. If you think about stubble management, in theory, if we all ploughed and buried the stubble, we would actually reduce the amount of ascospore production, but the effect would be relatively small, and pretty much everybody would have to do it in order to have an effect, because of the effective long distance dispersal, and we would burn an awful lot of diesel to achieve that. Whereas for yellow rust, the main source of the epidemic is really on your own farm, and if you can manage the stubble in a way that reduces the green bridge, not necessarily burying it, but making sure there isn't green tissue, can actually have quite a useful effect. And similarly for yellow rust, we have quite effective major gene seedling resistance in some varieties, and that seedling resistance can be so effective that it reduces the initial inoculum, whereas the level of seedling resistance against septoria in current varieties is not really sufficiently powerful to have that type of effect. But fortunately, the variety resistance we have against both septoria and yellow rust later on in crop growth is sufficiently effective to reduce the epidemic growth rate. And similarly, we have really effective fungicides against both pathogens. And then finally, our ability to reduce carrying capacity. We really don't have much option for reducing septoria by managing our nitrogen fertilizer. And I've put a kind of maybe for yellow rust. Certainly it's worthwhile avoiding excessive nitrogen uptake, which may be a combination of both the release of soil mineral nitrogen from preceding crops and the application of bag nitrogen. But to really constrain the epidemics to a very low level, we'd need to reduce the amount of nitrogen below the economic optimum. So effectively, we need to manage the nitrogen carefully to avoid excess and try to hit that optimum level. We can't really, I don't think, be constraining our yield potential by reducing the nitrogen level below that. So to conclude, it's most effective if we can control epidemics by a combination of delay, slowing the epidemic and limiting the carrying capacity. We need to match the control options to the pathogen, and we'll cover the control methods that are available to us in more practical detail in later videos. So thank you for watching.